Okay, this is lecture 4 in the cell pathology stream of the cell pathology and infection module, and this is a lecture looking at the genetic changes leading to cancer formation. So cancer is a disease which is characterized by the accumulation of genetic mutations in the cancer cells, and those mutations are not present in the patient's normal cells. So these are what we call somatic mutations. So these mutations accumulate over time. They accumulate in specific genes that control growth, cell death, survival, immortality, that type of thing. And we're going to go through the sorts of genes and the sorts of processes that are affected by these genetic mutations. It is fair to say that cancers do not form unless there are permanent genetic mutations in the tumour cells themselves. Now that doesn't mean that cancers are not hereditary. Cancers can be hereditary, but those mutations are different. Those mutations are germline mutations present, passed on from one generation to the next that predispose to cancer, and then subsequent somatic mutations drive cancer formation. What we're looking at here are the somatic mutations that occur irrespective of what the initial cause is. And in a later lecture dedicated to tumour suppressor genes, I will cover some of those hereditary mutations that cause inherited cancer syndromes. So much of what I'm talking about today is covered in this landmark summary review called the 6 Hallmarks of Cancer, which is now quite old. There is a new version of it if you click on this link here, and you can read about the 10 hallmarks of cancer. What I'm going to focus on are the original 6 hallmarks of cancer and how genetic changes change the cell to address these 6 prerequisites for cancer formation in solid tumours. And these are the 6 hallmarks, and you may well have covered them briefly in another module. What I'm going to do in this session and in subsequent sessions is go through some of these pathways and show exactly how specific mutations alter the cell's behaviour so that it achieves these 6 hallmarks of cancer. So self-sufficiency of growth signals, this is the epidermal growth factor receptor signaling. It can be RAS and RAF mutations. We're going to cover these in the oncogenes lecture. Wnt beta-catenin, I will cover that at some point as well, maybe in hereditary cancers. So this is effectively like a cell that's got its foot on the accelerator pedal. This mutation is the inability to put your foot on the brake pedal. This is insensitivity to anti-growth signals, and we have something called transforming growth factor beta. That's an anti-proliferative growth factor which stops cells from proliferating. So if cells cannot adequately signal this growth factor, they cannot stop their growth when they're supposed to. This is also linked into the PRB or retinoblastoma protein axis, which I will mention today, and I will mention again in the tumour suppressor genes lecture. I'll also throw in P16 as well. That's a protein that can stop the cell cycle. I will briefly cover cell adhesion and proteolysis. So this is related to tissue invasion and metastasis, because for a cancer to be classed as a cancer, it has to break through basement membranes, enter a different tissue layer, so be called locally invasive, and then to be metastatic it has to achieve all of the features of the metastatic cascade, which I'll talk about in this lecture, the ability to spread throughout the blood or lymphatic system. I will cover telomerase today. So telomerase is an enzyme which maintains the ends of chromosomes, which naturally get eroded every time a cell divides, and if these telomeres get too short, then the cell undergoes crisis and cell cycle arrest. So we'll look at how tumour cells can become immortal and allow them to divide more times than a normal cell. I might not talk too much about the VEGF axis, that's the vascular endothelial growth factor axis, but this is just angiogenesis, and tumours need to be able to promote their own blood vessels, so vascular endothelial growth factor coming from tumour cells can drive new blood vessel formation. And then finally there's apoptosis. Apoptosis is programmed cell death, and cancer cells are very good at evading programmed cell death. So the P53 axis, that feeds into cell cycle as well, as we'll see later and in the tumour suppressor genes lecture later on in the module. The P53 detects or senses or helps to pass on signals from DNA damage, pass on signals of generalized cell stress, promotes cells to undergo apoptosis, also promotes cell cycle arrest. So a tumour cell will effectively refuse to undergo cell death when it should, and refuse to stop growing when it should. And it's the p53, which is going to be the main part of this cluster of genes here. It's p53 that I will mostly talk about on this module. Now, depending on what your previous knowledge of the cell cycle is, I've put a separate video on Blackboard, which is an introduction to the cell cycle. It's one that I deliver to the second years, and this covers phases of the cell cycle and covers many of the things that I'm going to talk about in this lecture that you might not have come across before, or you may have come across relatively briefly in PBOD last year. This is things like cyclins, cyclin-dependent kinases, CDK inhibitors. So I've got separate lectures which are supplementary, which I'll put on Blackboard with an introduction to the cell cycle to remind you of what you learned in PBOD. And I've also got a lecture which is the introduction to p53. If you want to watch that now, that's fine. I will cover p53 in a bit more detail in lecture 5 or 6, but I've got a specific supplementary lecture on p53 if you want to get your head around it now, based on the couple of slides I'm going to talk about today. So those are both going to be under this week's folder, but I will highlight them as supplementary if you wish to look at them. So one thing to be really clear about is that cancer acquires due to a series of genetic changes that distinguish them from normal cells. So a cancer cell originates from what used to be a normal cell. That normal cell accumulates one mutation which makes it behave abnormally, usually hyperproliferative or immortal or something else on the hallmarks of cancer. And then a fairly predictable sequence of mutations occur to then change that cell into a malignant cancerous cell. And this is just shown here for colon cancer. This is what's known in the trade as the Vogelstein model, because this was figured out back in the early 90s by Vogelstein, and it's been added to since that early landmark study. But this illustrates the sorts of changes that are happening to a normal cell, and the initial insult to the cell can come from many different places. And we'll start on the top row. So this is your normal colon epithelium. The first hit is typically a somatic mutation in an otherwise normal cell in the APC gene. Now the APC gene is adenomatosis polyposis coli. That is part of the Wnt signaling pathway, which I will only briefly mention in this module. It's not a pathway that you're expected to know much about. That gene becomes inactivated, or one copy of that gene becomes inactivated. Then subsequently we get a second copy of that gene inactivated, and this is just down to sheer bad luck and the combination of bad luck and high carcinogen burden. So we end up with one cell which has got two defective copies of the APC gene. That means that the cell is now APC null or APC defective. Okay, so the first hit happens, and then subsequently sometime later the second hit happens, and that kickstarts the process when you get an aberrant crypt in the colon. So this is altering a cell's differentiation and subsequent proliferation. We then tend to get some ill-defined genomic instability or possibly some epigenetic changes, and then we very reliably get a mutation in KRAS. I'm going to mention KRAS today, and you're going to hear about KRAS throughout this module. So it doesn't matter if you don't quite get KRAS today or know what it is. Then SMAD4 tends to become mutated or overexpressed, and this is related to the TGF-beta axis, which again I'm not going to talk about in any great detail. I am going to talk about p53. This addresses the apoptosis side of things, and only then do we have cells which are bona fide cancer cells. Up to this point they are adenomas. This is benign. This is non-invasive. Once we get the p53 mutations, we tend to rapidly then get progression to invasive cancer. Then subsequently we get other changes, usually changes to gene expression, and the cells will start to express matrix metalloproteinase, which I'll talk about today, and change their cell adhesion molecules so they can either become stickier or more slippery. They can slip away from their partner cells and interact with blood vessels and spread around the body. So on this Vogelstein model, we have got defects which affect ability to proliferate. We've got defects that affect ability to proliferate there. We have genomic instability here, which is a new hallmark of cancer. So the SMAD4 mutations is on the TGF-beta axis. We sometimes get the TGF-beta receptor itself mutated. We have got P53, which is addressing apoptosis. And then we've got the adhesion and MMPs, which are addressing the migration aspect. So if you just flick back to the hallmarks of cancer, what you can see is on the hallmarks of cancer, all of the genes that I'm talking about are addressing one of these hallmarks of cancer. Now the telomerase is not on the Vogelstein model, and the VGF axis is not on the Vogelstein model, but I can tell you where they will occur later on in the lecture. So many of these pathways that I've talked about you will cover at some point during this module, possibly with the exception of details of the Wnt signaling pathway, which is related to APC and beta-catenin here. Now, it's probably a good point to just mention, and this is relevant to the tumour suppressor genes lecture, an alternate way of kickstarting this whole process is a mutation of the APC gene which you inherit from one of your parents. And if you inherit that APC mutation from a parent, then every cell in your body is defective for one copy of APC, and then any one cell in your body can then get a second mutation on the other homologue of APC, and that will kickstart the process. So this is going to be covered in great detail in tumour suppressor genes lecture, and that's related to what we call Knudson's two-hit hypothesis, which I will cover in a later lecture on hereditary cancer syndromes. So first proper hallmark, we're going to look at self-sufficiency of growth signals, and this basically means that the cancer cell can produce its own growth signals. And one of the simplest ways of doing that is for the cancer cell to produce its own growth factor, which it then secretes and it acts back upon the cells. So normally a cell will respond to growth factors which are external to it. Some other cell has produced growth factor, such as platelet-derived growth factor. That is acting upon the cell, that is causing the cell to proliferate. Platelet-derived growth factor, it is a growth factor derived from platelets. Think about platelets, blood clotting, where you've got blood clotting occurring at the site of a wound. You secrete or release lots of platelet-derived growth factor, and that encourages wound healing. So it's a very potent growth factor. In cancer, the tumour cells themselves can produce their own platelet-derived growth factor, and they're not relying on an external source of that growth factor, therefore they will proliferate more readily. The signals from those exogenous growth factors go through growth factor receptors, and what the cancers can do is either overexpress their growth factor receptors so they are permanently switched on, or respond to lower levels of growth factor. For example, androgens are required by prostate cancer cells, so prostate cancers overexpress the androgen receptor. Breast cancers overexpress HER2, lung cancers overexpress the epidermal growth factor receptor. So this is something that cancers do. They amplify, as in they duplicate their genes that code for those growth factor receptors, so they respond to much lower levels. What you can also have is mutations to the growth factor receptors, so those growth factor receptors are permanently switched on, even in the absence of any growth factor binding. And again, androgen receptors can become mutant so that they no longer require androgens to bind. Epidermal growth factor receptor frequently becomes mutated in lung cancer, so that the crucial signaling domain is permanently switched on, and the cell thinks that there is growth factor outside the cell when there isn't. Self-sufficiency growth signals can also come downstream of the growth factor receptor, so ligand binds receptor signals via some intracellular intermediates. One of the key ones is RAS. RAS is a proto-oncogene, and I'll cover it in great detail in the oncogenes lecture. But RAS frequently picks up mutations which renders it permanently switched on. So you can have normal levels of growth factors and normal growth factor receptors outside the cell, but if RAS is mutated, RAS passes on proliferative signals and drives cell proliferation. And this is summarized here, where you can find all of the different defects in the proliferative pathway that can drive proliferation. And we will cover this pathway in far more detail in the oncogenes lecture, but for now, the tumour cells can overexpress the mitogen, any one of these growth factors. That mitogen binds to the receptor, so the receptor can either be overexpressed or mutated, so it's permanently switched on. So this activating signal part of the receptor creates the tyrosine kinase domain in these receptors. That can be permanently switched on. RAS, which is a signaling intermediate, can be permanently switched on, as can all of the kinases downstream from RAS. They can all be switched on. Their job is to switch on an oncogene or a proto-oncogene called MYC. MYC is a transcription factor, and the job of MYC is to drive expression of cyclin D. Now, as you heard in your PBOD lecture last year, cyclin D kickstarts the cell cycle. So MYC is an oncogene which is frequently overexpressed in certain cancer types, and the role of MYC normally is to switch on cyclin D in a tightly regulated manner. If MYC is amplified or overexpressed or translocated, and we'll come on to that in a later lecture, cyclin D is permanently switched on, and that will drive the cell cycle. Now, if you've forgotten all of your cell cycle, as shown here, G1S, G2 and M, and all of the control mechanisms that control the cell cycle, I am going to go through them briefly here, but there's a more extensive supplementary video on the cell cycle on the Blackboard site in this week's folder. So if you are unsure about the cell cycle, cyclin, CDKs, CDK inhibitors, retinoblastoma, take a look at that lecture, because I'll probably explain it in more detail than I will cover here, because those videos cover the level of detail that you heard in your PBOD lecture a year or two ago. But as a brief summary or reminder, this is the cell cycle, G1S, G2M phase. G1 is a part of the cell cycle where cells sense growth factors and effectively listen out to growth factors. This is also the period where TGF-beta is growth inhibitory to cells, and the growth factors are trying to drive the cell through G1 of the cell cycle to the R point, and the R point is controlled by PRB, which is the retinoblastoma protein. The cell cycle is controlled by cyclins, and as I showed you on the previous slide, with growth factor signaling, cyclin D is switched on. So at the start of cell cycle, like here, cyclin D is off. Cyclin D rises in response to growth factor signaling because MYC will switch it on, and then that will do something which I'll cover shortly, that will result in the switching on of the next cyclin, which is cyclin E, also called the G1S cyclin, because that's the bit of the cell cycle it controls. That switches on the S phase cyclin, which controls gene expression to make cells ready or functional in S phase, and then we've got the M phase cyclin, which controls metaphase to anaphase transition. Now, the job of these cyclins, so this cyclin D here and cyclin E here, is to bind to a cyclin-dependent kinase, or CDK. And these cyclin-dependent kinases will phosphorylate key proteins and change the behavior of the cell. One of the key proteins that CDKs that bind to cyclin D and E will bind to is the retinoblastoma protein. So the main one to consider is cyclin E, which is this one, will bind to some cyclin-dependent kinases, and those cyclin-dependent kinases will phosphorylate the retinoblastoma protein and allow the cell cycle to progress. And this is shown here. This is the cell cycle, and around the outside is a circle which illustrates the degree of phosphorylation of the retinoblastoma protein. And the retinoblastoma protein is called the guardian of the R point. You need to phosphorylate retinoblastoma protein to progress from G1 into S phase. So throughout G1, retinoblastoma protein is unphosphorylated and then hypophosphorylated, so low levels of phosphates are tagged on by cyclin D bound to CDK4 and 6, and CDK4 and 6 do that phosphorylation. We then get a rapid expression of cyclin E, which activates its cyclin-dependent kinase, CDK2, and that completely phosphorylates the retinoblastoma protein, so it changes its conformation and shape, and that changing shape is crucial in driving the cell through G1 phase into S phase. And then PRB remains highly phosphorylated throughout the cell cycle and then gets dephosphorylated at the end. And this slide explains what is going on at that junction at the R point. So in Early and mid G1, PRB is either unphosphorylated or hypophosphorylated. It might have got a few phosphorylations tagged on by CDK4. The cell has to then progress through the R point, and the only way it can progress through the R point is if CDK2 is activated by cyclin E, which is the next cyclin that is expressed after CDK4. The job of CDK2 is to phosphorylate PRB, which normally sequesters or holds onto the E2F family of transcription factors. And these transcription factors are crucial for driving new gene expression, which will drive S-phase genes. So genes that are required for S-phase are generally induced by the E2F transcription factors. So cyclin E becomes expressed, CDK2 then becomes active. CDK2, cyclin-dependent kinase, it's a kinase. The job of a kinase, all kinases, is to phosphorylate something, and that target here is the retinoblastoma protein. When it's phosphorylated, it changes its conformation. That releases the E2F transcription factors, and those E2F transcription factors then translocate to the nucleus and switch on crucial S-phase gene expression. Now, just as a reminder back to what we're actually talking about here, we're actually on the section of insensitivity to growth inhibitory signals and insensitivity to controlling the cell cycle. And what happens in cancer is anything that can make this process happen more efficiently tends to happen. One of many possible things, and one of the many things that can happen is the tumour cells lose the ability to make PRB protein because the RB1 gene that codes PRB is defective, it's acquired mutations. So loss of PRB due to RB1 gene deletion is a very common occurrence in cancer, and if RB1 protein doesn't exist, the E2F transcription factors are always free to initiate S-phase even when S-phase shouldn't occur. So that's why PRB mutations are so common in cancer. And the PRB protein is a target for many mechanisms of inactivation, all of which are going to drive tumour formation. And it's directly targeted by viral oncoproteins. And as we'll hear in a later lecture on human papillomavirus, the human papillomavirus expresses a protein called E7, and E7 targets hypophosphorylated retinoblastoma protein, inactivates it, and or sends it for degradation. So two different viruses, adenoviruses and HPV. The adenovirus has this oncogene. The SV40 has the large T antigen, and HPV has the E7 oncoprotein. All three of these viruses can modify the cell cycle by having their own viral proteins which disrupt or inactivate or degrade the retinoblastoma protein, therefore driving the cell through the cell cycle, which is what the virus wants the cell to do. So we can summarise how tumour cells can override the R point and then be either insensitive to antiproliferative growth factors, or they can be self-sufficiency for their own growth. And these work hand in hand. Sometimes they have mutations in one, sometimes mutations in another. So tumour cells can have mutations that inactivate the TGF-beta receptor, so therefore they no longer respond to the antiproliferative effects of TGF-beta. TGF-beta normally downregulates cyclin E. It can also upregulate some inhibitors to cyclin-dependent kinases. So that will drive proliferation. Tumours often overexpress cyclin E by gene amplification. That will also drive proliferation. Tumours frequently have gene deletions to the RB1 gene. The RB1 gene codes PRB, so that if you have no PRB protein, you have no ability to sequester E2F transcription factors. What's not on this list is E2F transcription factors themselves can be overexpressed, so there's more E2F, there's more E2F than PRB, and therefore the E2Fs are always free to drive S-phase. And then we can have viral oncogenesis. So SV40 in E7 oncogene we know interacts with PRB, and similarly adenovirus EIA can do something very, very similar. Although these are not particularly oncogenic in terms of cancer causing, they just disrupt the cell cycle. HPV is cancer causing because it also has something that disrupts p53, which we'll talk about very shortly. So to finish off, this is going to be the end of part 1 of this lecture. To summarise your knowledge so far, what I want you to do is write down a short paragraph to explain each of these individual points and put in as many signalling details that I've mentioned in this lecture so that you get a full understanding of all of these signalling pathways. So I'll let you do that, and then coming up next, or if you look on Blackboard, there will be a second video that finishes off the hallmarks of cancer.